All the reactions of cellular respiration that we just saw are made possible by the presence of enzymes. An enzyme is usually a protein. About 99% of all enzymes are made up of proteins. They are in their tertiary form with a very specific structure. Only about 1% of enzymes are made up of RNA. Enzymes carry out their function by speeding up chemical reactions. So we say that enzymes are catalysts. A catalyst is a substance that speeds up a reaction. The enzyme achieves this by lowering the activation energy required for a chemical reaction to begin. When chemical reactions begin, they need a large amount of energy to get them started. It's an injection of energy. The enzymes lower the amount of activation energy required by a chemical reaction, and in this way they speed up the reaction. An enzyme is highly specific to its substrate. So if you take lactase, for example, it is highly specific to the structure of lactose. So the enzyme and substrate actually fit into each other like a lock and key. And this is made possible because each enzyme has a business end. And this business end of the enzyme is called its active site. The shape of the active sites is highly specific. If an enzyme loses its shape, it also loses its function because the active site is disrupted and it can no longer recognize its substrate. This can happen if the temperature is too high or if the pH is too low, in order to understand the concept of activation energy and the role of an enzyme to reduce the activation energy to start a chemical reaction, let's take a look at the analogy of this guy here that needs to study. There is a huge barrier of procrastination that exists before he starts to study. So we'll call that the activation energy, the energy needed to get him started. Now, this barrier can be reduced if there is a catalyst. The catalyst can come in the form of a due date, an approaching exam, or a reminder from his instructor. That can actually reduce the barrier to procrastination and get him started. So notice here that the activation energy has actually gone down. The enzyme works in much the same way. Here the chemical reaction is the process of studying that you see here, that is shown by the book, and the enzyme or the catalyst lowers the activation energy required to start the reaction. So here we have it again. Enzymes lower the activation energy required to start a chemical reaction. It is a catalyst. The structure of an enzyme determines its function. The shape of an active site of the enzyme is extremely important because it matches the shape of the substrate. So the enzyme and substrate can fit together like a lock and key. Here is the active site of the enzyme, and here is the matching substrate. The enzyme is a protein and exists in the tertiary form. And if you remember, the tertiary structure of a protein is held together by weak bonds such as hydrogen bonds and R interactions. Now, these weak bonds can be easily disrupted by high temperature or low pH. If an enzyme is exposed to either low pH, which is acidic, or high temperature beyond 40 degrees Celsius, then these H bonds and R interactions are disrupted. And the enzyme loses its shape, and the active site loses its shape as well. So the enzyme can no longer recognize its substrate, and therefore it is no longer functional. We say that the enzyme is now denatured. Here is an enzymatic reaction between lactase and lactose. Here's lactase in yellow, and the structure in pink is lactose. We know that lactase is an enzyme because the word ends in the suffix ase. We know that lactose is a sugar because it ends with the suffix ose. Here is the active site of lactase. It matches perfectly with the substrate, which is lactose. Like we said earlier, the enzyme is highly specific to its substrate. What we have here is an enzyme substrate complex. There's the E for the enzyme and S for substrate. Here's where the actual speeding up of the reaction takes place, where the enzyme has converted the substrate lactose into the monosaccharides glucose and galactose. So those are the products in blue. Here's glucose, which is product 1, and galactose, which is product 2. Now the enzyme lactase is free. It is unaltered. Its active site is still the same. It can now be reused to repeat this reaction if necessary. This reaction will continue so long as lactose is present. So here we have it. Enzyme plus substrate gives you the enzyme substrate complex, which results in the enzyme plus product.